Now let's consider type III hypersensitivities or immune complex reactions. These reactions involve the interaction of soluble antigen with antibody and the deposition of the resulting complexes in the basement membranes of epithelial tissue. This hypersensitivity is pretty similar to type II hypersensitivity, because it does involve the production of IgG and IgM after repeated exposure to the antigens and the activation of the complement system. However, it differs from type II in that its antigens are not attached to the surface of a cell. They become free-floating complexes that float around through circulation and can be deposited in any of the tissues of fluid compartments. This causes an immune complex reaction. Let's look at how that works. After the initial exposure of a profuse amount of the antigen, the immune system produces large quantities of antibodies, and these antibodies circulate throughout the fluid compartments of the body. When the antigen enters the system for a second time, the antibodies that are floating around in each of the fluid compartments bind to the antigen and form complexes. As you can see here, antigen-antibody complexes. There are such large quantities of these that they're forced into the tissues and become lodged in the basement membranes. This summons inflammatory players like the neutrophils and complement proteins. Usually, the neutrophils would be able to eliminate the antigen-antibody complexes. However, because they're so numerous and they're trapped here in the basement membrane, they're inaccessible to the neutrophils. So the neutrophils release their lysosomal granules anyway, which now are going to digest the tissue and cause vast destruction of a number of different tissues. These tissues could be blood vessels, heart and lungs, the joints like we see in rheumatoid arthritis, the skin, as well as the kidney. Now, in the early days of immunotherapy, vaccines and serums were tested on animals. They were injected into their skin, and then any sorts of reactions were observed. Hypersensitivity reactions were really quite common. Not only anaphylaxis, but Arthus reaction and serum sicknesses were identified. Our Arthus reaction involved localized dermal injury due to inflamed blood vessels in the vicinity of an injected antigen. Usually it's a fairly acute response. It's localized and occurs after the second injection of a vaccine, like in the case of booster vaccines. The area becomes red, hot to touch, swollen, and extremely painful. All of this causes destruction of tissues around the vessels and summons all the mediators of inflammatory response, like histamine. Serum sickness, on the other hand, is a systemic injury, as opposed to the Arthus reaction, which remains localized at the injection site. In serum sickness, the injury is initiated by antigen-antibody complexes that circulate in the blood and settle into these membranes at various sites. So injection of animal hormones and drugs are the most common cause of serum sickness. The immune complexes enter circulation and they're deposited in the tissues. This is just like the figure that we just looked at, where they become embedded in tissues around the heart and kidney and skin, as well as the joints. Serum sickness was named for the condition that appeared in soldiers after they received repeated doses of horse serum that were used to treat tetanus. These days, though, serum sickness is at least partially responsible for several prominent autoimmune diseases, like lupus erythematosus, rheumatoid arthritis, Graves' disease, which is an immune reaction that causes oversecretion of thyroid hormone from the thyroid gland, and then myasthenia gravis, which is an autoimmune response to the nervous system. Both Arthus reaction and serum sickness are like anaphylaxis in that they require sensitization and preformed antibodies, but they're different because they depend on IgG, IgM, or IgA rather than just IgE. They require large doses of the antigen, and their symptoms are delayed. Let's take a look at an animation that shows how type III hypersensitivity works.
Immune complexes consist of antigens and antibodies bound together. Although large immune complexes are engulfed by phagocytes and removed, intermediate-sized immune complexes are not readily removed. These intermediate complexes react with and activate complement. For example, complement activation in blood vessels causes basophils to degranulate, which causes vasodilation. Immune complexes entrapped in blood vessels or other tissues, such as kidney glomeruli, activate complement, which attracts neutrophils and causes them to degranulate. Neutrophils release enzymes responsible for tissue damage.
So up until now, we've been talking about B cell involvement and antibodies. Now we'll cover type IV hypersensitivities, where we'll be concerned primarily with the T cell branch of the immune system. Symptoms arise in 1 to several days following the second contact with the antigen. This is why they're called delayed reactions. They result when T cells respond to antigens that are displayed on self-tissues or transplanted foreign cells. Typically, we'll see these in delayed allergic reactions to infectious agents, in contact dermatitis, and in graft rejection. Here's an example of the delayed infectious hypersensitivity. The tuberculin reaction. This is a typical test used to test for exposure to tuberculosis. It's a classic example of delayed type hypersensitivity. Basically what happens is a person who's been sensitized to tuberculosis is injected with a tuberculin extract. The inflammation begins about 24 to 48 hours after injection, and the response involves these helper T cells. After the dendritic cells have processed the allergens, they pass them to the T cells, and then these activated T cells release cytokines that recruit inflammatory cells like the macrophages, neurophages, and eosinophils, and they show up at the reaction site. In the figure on the right here, you can see a typical response from this delayed infectious allergy response. Any area of inflammation that's greater than 10 millimeters is considered to be a positive reaction. Let's take a look at an animation of type IV hypersensitivity response before we move on to look at some other specifics.
Harmful effects produced by the mechanisms of cell-mediated immunity are referred to as delayed hypersensitivity. Delayed hypersensitivity is mediated by Th1 cells, also called Td cells, and is responsible for contact dermatitis, tissue damage in a variety of infectious diseases, and rejection of tissue grafts. A contact hypersensitivity, such as poison oak dermatitis, is initiated by contact with the antigen, which is also called the allergen. During contact, a small molecule from the plant, called a hapten, binds to a carrier protein in the host. The carrier protein with bound hapten is ingested by a macrophage, and the hapten peptide is then presented on the surface of the macrophage on a class II MHC. Th1 cells with T-cell receptors capable of recognizing the hapten peptide antigen interact with the presented antigen, become activated, and increase in numbers. When the host comes in contact with the allergen a second time, the Th1 cells react with the hapten peptide being presented on the macrophage and release cytokines, resulting in attraction of more macrophages, followed by inflammation and skin lesions. Characteristic skin lesions appear after 24 hours, reaching their peak at 48 to 72 hours after the second exposure to the plant.
So the most common delayed allergic reaction is contact dermatitis, and as we just saw, it requires sensitizing before we see a provocative dose. That is, we need a second exposure in order to see this reaction. The allergens involved in this response are most commonly resins and poison ivy or poison oak, or other simple household articles like jewelry or cosmetics and certain drugs. Most of them involve a haptens, or a very small particle, that once it's inside the host, needs to be associated with another carrier molecule. But here we have the chemical antigens. As they make their way through the skin, they're met by these Langerhans cells or dendritic cells in the epidermis. They're taken in by those dendritic cells and then they're presented to the T cells. During subsequent exposure, these T cells will then attract lymphocytes and macrophages to the area. These will give up enzymes and inflammatory cytokines and damage the epidermis, thus causing blistering, which, if you've ever experienced it, you know is exceptionally itchy. Now let's take a look at how organ transplantation works. Transplanting and grafting are now relatively common practices. However, the lymphocytes are always on the lookout for foreign antigens, and they're ready to destroy. The bulk of damage in graft rejections involves cytotoxic T cells as well as other killer cells. The genetic and biochemical basis for graft rejection involves those MHC markers that we were talking about a couple of chapters ago. MHC markers, you'll recall, are important in recognizing self and regulating the immune response. Each person can exhibit a variability in the pattern of these cell surface MHC marker molecules. The more closely individuals are related, the more closely their MHC markers match. When transplants occur, the donor's tissue may display different MHC markers, and the T cells of the recipient will recognize its foreignness and react against it. The reaction can be a two-way process. You can either have the host rejecting the graft, or in the case of immunodeficiency, you may actually see the graft reject the host. When the cytotoxic T cells recognize foreign class 1 MHC markers on the grafted cells, they release interleukin-2. This causes a general immune mobilization and amplifies the helper and cytotoxic T cells that are specific to that foreign antigen that's on the donated cells. The cytotoxic T cells will bind to the grafted tissue and then secrete lymphokines. This begins the whole rejection process and causes antibodies to be formed against the graft and eventually destroys the vascular supply, causing imminent death of that graft. Now, the other side of the deal is that the graft could reject the host. Some grafted tissues contain passenger lymphocytes, and these lymphocytes will go into host tissue and in a similar way as I mentioned above, cause a rejection of the host. This can actually cause a much more severe situation called graft versus host disease, or GVHD, which means that any host bearing MHC markers that are foreign to the graft can be attacked. This can become a huge, widely systemic and toxic situation where GVHD resides in many organs throughout our system. Now, there are several different types of grafts. They're classified based on the genetic relationship of the donor and the recipient. Autografts make sense. This is tissue that's transplanted from one site on an individual's body to another site on his body. Skin replacement and burn therapy is a great example. Or often we use the veins in the lower leg in heart bypass surgeries. Isografts involve tissues from identical twins. Of course, here there are no foreign antigens, and thus there's no real risk of rejection. Allografts are where we see exchanges between genetically different individuals, but they belong to the same species. Of course, the objective is to match the donor and recipient to the most close genetic correlation possible. Allografts are the most common grafts that we see. Xenografts are a little bit less common. They're when we see a tissue exchange between individuals of different species. There are a couple of different types of transplants offered too. And here's a really interesting statistic. Back in 2007, there were 180,000 people that were living with functional livers, kidneys, or hearts from transplants. Pretty amazing statistic. Transplants have been performed on pretty much every organ, including parts of the brain, but the most frequent we see are skin, liver, heart, kidney, coronary artery, corneas, and bone marrow. Sometimes these come from live donors. Sometimes they come from deceased donors and even fetal donors. For example, fetal pancreases have been seen as a great potential treatment for diabetes, and fetal brain tissues as great treatments for things like Parkinson's disease. We've even seen parts of livers or lobes of lungs being transplanted from a live parent into their child in order to save them. And now, with the advent of so many great stem cell technologies, there are many, many more possibilities for treatment, including the storage of umbilical cord blood. Just think that umbilical cord blood contains stem cells that can be utilized by that child or any of its close relatives. However, one thing to consider is the use of stem cells in the United States has been severely restricted, and thus we're falling way behind in medical technologies. That brings us to the end of this section, but before we move on, let's have a quick concept check. What type of tissue transplant would be least likely to cause a host versus graft response? A xenograft, an allograft, an isograft, or an autograft? Well, I think there are two components here that are good possibilities. Either isograft or autograft. Isograft coming from twins, or autograft coming from self. Autograft, of course, is the least likely to have any kind of reaction. However, isograft is a very close follow.